working on sequencing and validation of single nucleotide polymorphism markers linked to carotenoids in cassava. Cassava is a plant which belongs to the family Euphorbiaceae, and it is a species is Manihot esculenta. In fact, this is the only species in this family that is edible. It's commonly called cassava or manioc or yucca. As you can see in the picture, cassava is mainly cultivated for its tuberous roots. Okay, most people cultivate cassava for the edible roots, although the leaves can also be used as a vegetable, which is not often used depending on the variety. Globally, cassava is produced, and Nigeria is actually the highest producer of cassava, with more than 50 million metric tons, according to FOA stats in 2020. Cassava itself is a perennial woody shrub that is clonally propagated and has the ability to tolerate droughts and low soil fertility. That is why it is mostly cultivated by poorer communities, because it can be used as a food security crop that is capable of averting hunger, during the drought conditions. The cassava plant is widely produced and is actually the fourth largest stable food commodity globally after wheat, rice, and maize. Okay, it is a major staple food commodity in sub-Saharan Africa, and studies by Oyemawa shows that about 84% of households in Nigeria actually consume a form of cassava daily. Notwithstanding the high rate of consumption of this crop, it is actually deficient in some micronutrients, although it is rich in carbohydrates and a little protein. And these micronutrients include zinc, iron, and vitamin A. Okay, and most cassava varieties are white, although some are off-white. And in these studies, we are actually interested in the vitamin A component, seeing how to increase the vitamin A in cassava varieties. And vitamin A is crucial for good vision and proper functioning of the immune system, especially among preschool children, pregnant, and lactating mothers. In fact, the deficiency has recently become of serious public health concern, okay, because it leads to childhood blindness, reduced immune function, and increased risks of mortality. Okay, this vitamin A is actually gotten from carotenoids in plants. Carotenoids are the precursors of vitamin A in the body. It occurs actually as strongly colored yellow to red pigments found in plants, example in carrots and sweet potato. Okay, and this, naturally in the gene pool, cassava have some varieties that are yellow in color, although most of them have tuberous roots that are white. Okay, like we can see here in the picture, have some tubers that are white, that's number B. Some are C, cream colored, while some are A, very deep yellow. And it was discovered that those with the cream color and the yellow coloration have some amount of vitamin A. So we are now looking at, okay, how do we improve on the quantity of vitamin A in cassava due to the fact that it is, widely consumed in the community, so as to avoid the problems that would come as a result of deficiency of these vitamins. And it was discovered that the deep yellow cassava varieties are high in vitamin A, but low in starch content. So there is a need for breeding so as to improve on the edibility of this crop. So we carried out this study because cassava is a major staple food crop in Africa. So we saw that biofortifying cassava with provitamin A will have a significant positive impact on nutrition and overall health, especially among poorer communities. So the biofortification of cassava is ongoing through breeding, plant breeding, using the process of recurrent selection and recombination method, okay, which this process involves long breeding cycles. So the aim of this study was to develop molecular markers that can be used in the breeding process to facilitate the breeding, the release of varieties that are actually high in vitamin A. And you know, in the cassava breeding cycle, it takes up to more than 6 years to release a variety. And as you can see from the chart, in the seedling nursery stage, you know, a lot of samples are being sampled. About 25,000 to 100,000 seedlings are grown, after which selection takes place to advance selected progenies that are favorable to the clonal evaluation stage, where 500 to 3,000 clones are usually raised. Okay, at this stage, selection again takes place, you know, for those that are high in carotenoids and other agronomic characteristics, and they are advanced to the preliminary yield trial stage, where about 100 to 200 are also advanced. So at this stage, where we have to work with a lot of seedlings, applying molecular markers to identify carotenoid-rich seedlings would actually be faster. You know, it will save time for the breeders, and it will save costs, you know, of employing labor to carry out manual selections and evaluation strategies. So that's why we undertook this research to identify molecular markers that can be applied directly to the breeding process to facilitate selection and then quick release of vitamin A cassava varieties. Biofortification of cassava with provitamin A can be effectively achieved through plant breeding aided by molecular markers on target genes to increase selection efficiency. And this can be achieved through association studies. In this work, we did candidate gene association studies, and these can actually reveal key single nucleotide polymorphisms among the candidate genes that contribute significantly to accumulation of provitamin A in plants. And research have shown that some of these candidate genes in the biosynthetic pathway of carotenoids have actually played key roles, you know, in increasing levels of carotenoids in plants. They include the lycopene beta cyclase gene, the lycopene epsilon cyclase, the phytoin synthase gene, and then the beta carotene hydroxylase gene. Association analysis actually brings together, can be carried out in two ways, either the genome-wide association mapping or the candidate gene association mapping. Okay, in the genome-wide association mapping, the DNA is actually extracted and then subjected, you know, sequencing is done on the genome. While for the candidate genes are identified, that is, genes that are responsible or key to a particular function are identified and then RNA extracted, and nucleotide polymorphisms, you know, looked out for. These polymorphisms that may be associated with the phenotype. And then both strategies utilize population structure and kinship to identify phenotypes that are associated with key polymorphisms. In our article, using a candidate gene association approach, we looked out to identify markers within some genes that are associated with carotenoids. And for the methods, we utilized 4 candidate genes, which were sequenced from 167 cassava accessions. They include the beta-carotene hydroxylase gene, the phytoin synthase gene, the lycopene epsilon cyclase, and lycopene beta cyclase genes, which have been previously identified by other researchers. And then a total carotenoid content was determined using spectrophotometer, and The total beta carotene was quantified using high performance liquid chromatography for the 167 cassava accessions. The storage root yellowness due to carotenoid pigmentation was assessed using the color chart. Okay, this was done indirectly, indirect carotenoid assessment by color chart on a rate of 1 to 6 color rating, and then direct quantification by spectrophotometer and then high performance liquid chromatography to determine total beta carotene in the storage roots. Okay, so this is the chart which was used to determine the color. The 1 was for white, we scored 2 when the root was light cream, we scored 3 when the root was cream in color, and we scored light yellow when the root was 4, and 5 when yellow, and 6 when deep yellow. We also carried out candidate gene association analysis to count, which are data counts for the population structure and kinship using genome-wide single nucleotide polymorphisms generated through genotyping by sequencing approach. After the analysis, we identified 6 significant single nucleotide polymorphisms, that which we eventually used to design primers, called competitive allele-specific PCR assay. And this assay was used to validate the larger populations to see its association with the breeding population. Our results: candidate gene sequencing of the genes for beta-carotene hydroxylase, phytoin synthase 2 gene, lycopene epsilon cyclase, and lycopene beta cyclase yielded a total of 37 SNPs. Okay, and the total carotenoid content, total beta carotene, and color parameters were found to be significantly associated with the markers or single nucleotide polymorphisms in the phytoin synthase 2 gene, while the single nucleotide polymorphisms that were observed that were gotten from the lycopene epsilon cyclase were significantly associated with color parameters. The genes for lycopene beta cyclase and beta carotene hydroxylase were not found to be significantly associated with carotenoids or the color parameters. This table shows, the length of the genes that were extracted for the 4 carotenoid genes. For the phytoin synthase 2 gene, we discovered a total of 6 single nucleotide polymorphisms, while for the lycopene beta cyclase and epsilon cyclases, we discovered a total of 13, while for beta carotene hydroxylase gene, we discovered a total of 5 polymorphisms. After the association analysis, it was discovered that polymorphism in the PSY2, which is the phytoin synthase 2 gene, at the 572 position had the SNP, A and C SNPs, which actually caused an amino acid substitution, from alanine to aspartic acid, producing a total of 79%, explaining 79% of total phenotypic variation for pulp color. Okay, and after this analysis, a total of 6 single nucleotide polymorphisms were used to design molecular markers. And these same SNPs are the PSY2 gene. This is the sequence chromatogram after Sanger sequencing. And this is an alignment in bioedit with the reference sequence. Here is the reference sequence. And the dots are used to show consensus sequences so as to reveal the polymorphisms. Okay, these are the polymorphisms that we got for, in this position, 400, 540, 49. Okay, these accessions are actually white, while these are yellow. It was discovered that most of the white varieties had the T nucleotides, while the yellow accessions had C nucleotide. Upon conversion to amino acids, The first polymorphism, there was synonymous. It did not cause, there was no amino acid exchange. But the second for the PSY2 572 nucleotide had amino acid exchange, where most of the white varieties had this alanine nucleotide, while the yellow varieties had this aspartic acid. Okay, the 6 molecular markers that showed significant association with carotenoid content or yellowness were used to perform a validation, and we observed that the phytosynthetase 2 gene, which caused that as non-synonymous amino acid exchange, actually went ahead to had a 75, was able to explain 75% of total phenotypic variation for carotenoids based on the color chart. And this is quite remarkable, as this very marker can actually be used for, can be deployed into breeding programs to increase, carotenoids in cassava. In conclusion, cassava is an important stable food crop, for livelihood and food security in Africa. So to alleviate provitamin A deficiency, where cassava is a main staple, the development of improved cultivars with high levels of provitamin A is a major breeding goal in sub-Saharan Africa. Deployment of molecular markers in the breeding pipeline can increase efficiency and accelerate the rate of genetic improvement with respect to provitamin A biofortification, especially when this is combined with marker-assisted selection for other agronomic traits like resistance to virus diseases and also increase in dry matter content. Thus, the use of functional markers will allow breeders to pre-select the best candidates that combine desired characteristics for subsequent field evaluations. This work is actually the pioneer work for efforts to increase carotenoid content in cassava. And this is the first time molecular tools will be deployed to the breeding process to actually breed and release carotenoid-rich varieties to alleviate the menace of vitamin A deficiency in the continent. I want to appreciate my co-authors for our article, Candidate Gene Sequencing and Validation of SNP markers linked to carotenoids in cassava, the members of staff for the department, the Harvest Plus and Cassava Breeding Program for sponsoring the research, and also the Bioscience Center in Ibadan. Thank you for listening.